The last presentation of this session is on radiomics, pipelines, and the cancer data ecosystem delivered by Dr. Sandy Napel.
So thank you, boa tarde and muito obrigado for inviting me to speak at this meeting in this wonderful town that I've never been to before, and I find it very beautiful. And thanks to all the organizers for helping me to get here, and to Fred and others for inviting me. So I'm in the enviable position of being the last talk of the day. I'm between you and your cocktails. And also being the last talk of the day, I've found that a lot of what I was going to allude to and talk about has already been said. So with that, let's go drink. I will try to go quickly, and I'll try to just concentrate on the parts where I think I can add a little bit to all of the brilliant discussion we've already had. So to begin, I have to, of course, thank my team for helping me in everything that I do. It's a small, but it's a mighty group, and thank the funding agencies for helping us get the results that we do get. So a lot of this work that I've been involved in is supported by the NCI, the Quantitative Imaging Network. It's how Fred and I rediscovered each other. We actually knew each other many years before that, and been doing all of this work, particularly in radiomics and radiogenomics with the QIN. In this editorial that came out, I believe, in 2016, Bob Gillies and Hedy Resac basically brought radiomics to radiology. It had been done years before that by people in Europe and by various other people, but it became something that could be discussed among radiologists after this article appeared in Radiology. When we got involved in it a couple of years before that, it was in looking at lung cancer and trying to build radiogenomic models of lung cancer. And I just wanted to start by using this study that we did to sort of set the context for the methods that we talk about today. So basically with radiomics and radiogenomics, you start out with a bunch of images and you characterize them in various ways, either with computational features or with semantic features or both. They typically can get clustered and eventually produce a matrix where the patients might be columns and the rows are image features. You might also have excised tumors that have come out following the imaging exam. They can be processed, and all sorts of chemical assays, including genomics and all of the omics that can be done from tissue, go into a similar clustering pipeline, winding up with columns of patients and rows of features from the omics analysis. And why is this done? It's done because what you'd really like to do is discover, as we all know at this point, relationships between the image features. Let's see what I can do about that. Between the image features and the information in the chemical assays, and to discover those associations. It's not that one causes the other or vice versa, but the associations are very interesting and important. And if you combine that with EMR data, other clinical data from the patient, the discovery that has been done can be used to test models that can predict survival, response to therapy, et cetera. So this diagram really is an overview of the complete radiomics workflow, and it includes conventional radiomics as well as the deep learning approach. Most of my work has been above the line, sort of up here with engineered features, and that's pretty much what I'm going to talk about today, but I'll get back to this in a minute, this alternate pathway, which is through the deep learning architecture. So we'll start with tumor identification and annotating semantic features. Daniel Rubin and his group at Stanford have developed this software package which we use, which is called EPAD, and it allowed us in our experiments to identify tumors here illustrated in a lung cancer by putting some geometric object on it. But the important part that he was interested at that point, this is before we started thinking about computational features, was human annotated features. It was impossible to go into a radiology report and extract semantic features in those images because of differences in vocabulary, differences in the way people talk, hedges in reports, all these sorts of things. And so what Daniel and his group came up with was a way to use a controlled vocabulary to force responses so that these semantic features, and in this particular template that was developed for lung cancer by Ann Leung at Stanford, is to annotate several different features, including where is the anatomic location, and what is the nodule attenuation like in terms of verbal characterization, etc. It can also be attached to segmentations. We'll talk more about segmentations a little bit later. They're very important. And so it can be 2D or it can be 3D as shown here. And the semantic, even though we haven't talked really about semantic annotations very much today, they're very important, and there have been a number of papers that have been able to come up with very simple decision trees just based on semantic features. Here, just using 4 semantic features in lung cancer, one's able to predict very accurately whether or not the patient expresses the EGFR mutation. And this one, which comes out of combined efforts from UCLA and other institutions, looked at HCC, and with a very simple model based on semantic annotations, able to in a fairly accurate way, predict whether or not from images a patient's HCC has microvascular invasion, which some of you may know is very important to know, and right now you can only determine at pathology, and so it may already be too late at that point. But that was just to illustrate semantic features and to not forget them. The conventional radiomics approach, as you've seen, I'll go through this very quickly, there are all these different engineered features that can be computed upon the images. They're stored in a radiomic feature vector, and they go into a classifier, which combined with clinical data produces some sort of output. And you go through these, like I said, very quickly. Their size of shape, size features, how big is the object, what's its shape like, what is the edge sharpness as you go from inside the tumor to outside the tumor, the statistics of those as you go all the way around, and texture features. And as usual, it's illustrated here for 2D, but of course this is a 3D operation. Shape feature can be described in various ways. We've already seen examples of sphericity. There are other distance signatures that tell you things like how undulating the shape is, and these can all get packed into a feature vector as well. The edge feature vector tells you how sharp the tumor is with respect to the tissue that it's embedded in. And computational features, of course, include all the statistics of the gray values inside the tumor, all the histogram features, mean, minimum, max, etc. And finally, texture features. There are a number of different ways to compute texture features. This is one that was proposed by Adrien de Persange from Switzerland and published in this journal in 2010, which uses a bunch of filters to characterize the spatial distribution of gray values in the images. And there are many others, including Gabor features, Laws features, Haralick, etc. So, getting to be a little bit more practical now and how this talk fits into the cancer data ecosystem, we, with the funding of the NCI, have begun, and we're about 3-4 years down the road in producing what we call the Stanford Quantitative Image Feature Pipeline. And this, as you'll see, allows all sorts of exploration and discovery about what you can do with features and what you can learn from them. It's Linux server-based. It accepts images and segmentations. It accepts images and segmentations, DICOM images and DICOM segmentations. We also now have a module that accepts segmentations of other types, for instance, NIFTY from the neuro community, etc. It also accepts clinical data, which can be used for building predictive models, or those clinical features can also be part of the model building. And it computes radiomics features as well as building predictive models. Looks very simple, and in some ways it is very simple. And it comes loaded with Stanford's feature engine. This was developed in MATLAB by one of my students who became a postdoc later, It was published in Journal of Digital Imaging, and it computes hundreds of the many image features that we've talked about, shape, size, etc. That's available, and it comes with the system. In addition, there are user feature engines that can be uploaded to the system by any user as long as they're containerized. And we've standardized on the Docker container. And so if somebody wants to use a different set of features, for instance, Radiomix from the Stanford group, they can be Dockerized, uploaded to this platform, and those features can be used instead of the Stanford features or in addition to the Stanford features. We do have Radiomix and a few other ones such as SIFT and Delta features and things like that. We also have a very simple machine learning engine. It's been mentioned before in the meeting. It's just lasso, a sparse regression. We also will be supporting support vector machines. This code was actually written in R as opposed to MATLAB. So we really, as long as it's Dockerized, this platform can take code that was written in any language and in any operating system. And users can also upload their own machine learning engines as long as they're in Docker containers. And for instance, although we have not implemented this yet, it's perfectly feasible and in fact quite easy to take a trained deep learning engine and upload it in a Docker container and have it actually do a bunch of data testing using this platform. So I'm going to show you some screenshots just to give you the capability of what we've been able to do so far. One of the important design criteria was to enable it to directly connect with data structures and databases. So for instance, I've outlined the TCIA button. This is the Cancer Imaging Archive that Fred Pryor just talked about, and it will directly, using a REST interface, connect to TCIA, show you all the cohorts that it has, and you can select any one of those cohorts and run it through some of the workflows that I'm about to show you. You can also, if you just concentrate over here still, connect to EPADs that have image data in DICOM format as well as segmentations, or you can have your own local databases as well. So here's an example of connecting to one of the EPAD instances we have at Stanford. Here is a PET SUV cohort that was developed by one of my postdocs. Here are the image data sets that are available. These don't look de-identified, but they are. And what you can do once you pick a cohort is you can click the Run a Workflow button, and you'll get a pop-up which says, well, what do you want to do? And we have a bunch of pre-stored workflows. One that you're seeing here is all the workflows that include the use of the Radiomix 3D feature engine. There are DICOM versions for DICOM inputs, and another set of workflows that will accept Nifty inputs. And so, for instance, if you pick this, you can pick one of these workflows, and you'll see in a few minutes what the workflow looks like to run on the data set that you've picked. We also, the one I'm going to illustrate is using the Stanford feature engine, and it is this one, which uses the quantitative image feature MATLAB engine, followed by a lasso training machine learning model and testing after that. And so what that workflow looks like is the following. This will then pop up on the screen, and you can see all the inputs, DICOM images, clinical data, configuration files, and what the target feature is that you're trying to predict. Here's the workflow. The data gets set up, goes into the feature engine. They get combined and encoded with clinical data and semantic features if they're available, followed by a machine learning algorithm to do a sparse regression, and an optional step of taking different data and testing the model that you came up with in this step. So just an example of what some of the outputs are. So from the quantitative image feature engine, or from Radiomix, which I won't show. Here's from the quantitative image feature engine. You get this huge spreadsheet, image features in rows, patients in columns, and the first few here you can see are intensity features, so some histogram features. Moving down the list, we followed by shape features that were computed for the patients that you see in the columns, and these are all a whole bunch of different shape features that go into the shape feature vector. Following that, we get into a very simple shape feature, sphericity, which was mentioned here today, different sizes. Finally, the texture features, which are GLCM features in this implementation. And then once that feature vector, the feature matrix, where the columns are patients and the rows are features, are produced, you can download that feature vector and do whatever you like with it, or you can move it forward in the pipeline into a machine learning module, and in this case we had picked the lasso module, and so the output of the lasso module might look like something like this. Here is the small number of relatively uncorrelated and important features that were picked by lasso, put together in a linear model with these coefficients, and here's your model that you can use, for instance, to test on a completely different cohort. It also outputs the training performance in form of a ROC curve, so you can see how well this machine learning model was able to predict your clinical variable, when in this case was predicting survival from Radiomics derived from PET scans. So I skipped this whole issue here, which is segment. And I've heard a lot about segmentation today, and in my career I've been involved in segmentation and I've heard much about it. And so why did I skip that step? I skipped that step because as much as we make progress in segmentation, it is still a very difficult and open research area. And the wonderful talk by my new colleague from Microsoft, tremendous effort is being done, and I like the idea of these cloud systems that developers can use to develop and work on their own segmentation algorithms, because I don't believe anybody's ever going to develop a segmentation object that even works for one clinical problem forever. So I like the idea of users being able to develop them as they need them, and I congratulate you for putting that together. But I wanted to make some comments about segmentation. It's non-trivial, as I've said. Every lesion type has a different segmentation algorithm. Every modality requires different segmentation algorithms. Every instance, even within a modality, from different vendors or from different protocol acquisitions, requires a lot of work and tweaking. These are never perfect, and they almost always, particularly for radiomics, where you'd like to do this in a at scale, you'd like to do massive computations of radiomics, you really have to inspect the segmentations in order to know that you want to believe the features that come out. So here's an example of some work that I had the pleasure of doing with Jay Shariko, Pathy Kramer, and some others in the quantitative imaging network several years ago. We used a standard data set, and we had 3 different universities that had 3 different segmentation algorithms, and they each ran we each ran those segmentation algorithms 3 different times with different initial conditions. Here's an example of non-small cell lung cancer, 3 different algorithms, algorithm 1, 2, and 3, all get 3 different answers on this part solid, part ground glass nodule. And this is really only from 1 of the 3. You can see these don't agree very well, and therefore the downstream radiomics are going to be problematic depending on whose segmentation algorithm you used. Now let's consider the features that we extract from these segmentations. As I mentioned, size, shape, edge sharpness, histogram, texture, semantic features. Let's look at this in a sort of what the US would call a consumer reports format. Which ones of these are sensitive to segmentations and which ones are not? Certainly, these segmentations are all different sizes, they have all different shapes, and because the edge is in a different place, if you want to characterize your edge sharpness estimate from these, you're not going to be very happy about it. However, there are some features that may be a little bit more forgiving. The histogram features, These two probably agree very well because there is some ground glass that is subtended by this. This one just got the solid component, but certainly going to do a little bit better than any of these features did. Textures might be very well conserved across these segmentations, and certainly the semantic features don't use segmentations at all, and so there's no problem with them either. So let's consider what these thoughts might leave us with. So if you look at the Radiomix features, in our implementation, there's roughly 10 to 50 size features. There are that many shape features. There are about 100 edge sharpness features, histogram features, etc. Lots and lots of texture features. And many of these are highly correlated. I mean, as you've heard today, and as I mentioned, using lasso reduces the features down to a very small set because many of them are highly correlated, and it's not important to use all of them. So given that they're highly correlated, what's sort of a leap that you can make? Well, if they're going to be filtered down by something like lasso or some other feature reduction method, you're going to wind up with a model that uses fewer, less correlated features than you start out with over here. And so if you go back to the consumer reports, maybe we don't need these features. Maybe size is correlated with some texture feature because there is some biology that is causing growth and causing texture in a given clinical application. So maybe we don't need some of these features, and in some cases we therefore may find that we don't really need an accurate segmentation. So I'm encouraging people in my lab and people that I work with to look for these applications where inaccurate segmentation will not necessarily cause you to not be able to come up with a good model. But if you insist on segmentation, the quantitative image feature pipeline will accept a segmentation algorithm in a Docker container, and it can be configured in a workflow such as the one shown here. This is a segmentation algorithm that comes out of the Harvard and Brigham groups from Boston. So there are other challenges, and I'll go through these quickly. First of all, there is a lack of agreement on feature implementations and their names. What I call a certain texture feature, you might know by another name. Or what we both know by the same name, we may be computing differently. And so we have to worry about these sorts of things as well. One good effort in this direction comes out of Europe. It's the Image Biomarker Standardization Initiative. They're constantly updating their preprint, and they're trying to get the community to agree on nomenclature, on definitions, and also benchmarks for image processing so that we can believe that if we call an algorithm X, that when you compute algorithm X, you get the same answer as I do when I compute algorithm X. This is a good group to watch and be aware of as we move into the future. We've talked about some of these. I'll go very quickly. These are just some examples of how different software packages can result in different feature computations. Errors in this particular study from Larry Pierce from the University of Washington showed that there were errors in Radiomix values ranging from -2% to 100%, and all they were computing in this one was SUV max. So it was really a very interesting study. As we've heard, noise, slice thickness, reconstruction algorithms, dose, when you're using PET, just normal stochastic variation, patient gets on the scanner, gets off, gets on again, can all contribute to variance in these radiomic parameters. And these need to be, as we heard in an earlier talk today, filtered out as the ones that are not robust and don't get to move forward into the model building. This is a good effort coming out of MD Anderson, and that is they've built a sort of Radiomix phantom that has several layers of cork that can be brought from scanner to scanner, and the gray values that you get in the different scanners can be used to actually calibrate features so that if you scan on a GE scanner with a certain protocol and another member of your consortium is using a Siemens scanner with a certain protocol, that you might actually, by scanning a phantom first and doing some calibrations, you might be able to harmonize those acquisitions and use them. And that's exactly what they found in this paper. I just want to, as I close, I want to show the AI workflow in this diagram. There are basically two choices. You can go through this, before I only went through this way, but you can identify the tumor, segment it, and then go into the deep learning engine. The deep learning engine, just like engineered features, results in a vector of features, except these have been learned and not constructed. Or the other approach is to actually skip segmentation in the first place, because as you all know at this point, there are lots of publications coming out saying that maybe deep learning doesn't need to segment, or maybe deep learning will do the segmentation. So this problematic pathway may cease to be as big a problem in the future. And of course, they can be combined. And we've seen a number of articles come out recently where conventional radiomics features have been combined with learned features to go into a classifier and produce a result. And I mentioned this before. So in summary, in the minus 18 seconds I have, radiomics is advancing. We've seen many, many publications come out since the early 2010s. There are many software implementations and toolkits and pipelines available, including the one I showed you here. Radiomics faces lots of challenges. The features are sensitive to noise, segmentation, the heterogeneity of the imaging data, and of course, all of these problems are going to cause similar problems for the AI or deep learning approaches, and all have to be thought of as we move into the future. Just wanted to point you to an article that goes over all of the issues with whether or not radiomics has a future. You can read the arguments of why it is the future and why it isn't, and it was kind of a fun counterpoint to bring up all the issues that we deal with every day in this kind of research. So I hope I've shown you that pipelines are useful things. They can facilitate connection to stored testing and training data. The data can be public, like for instance, there's a direct connection to TCIA, clinical data to TCGA, and it will even connect to your own packs to pull your private data collections. It allows you to assemble a variety of different tools. You can build them yourselves in your own research group, or the community can contribute them for group use. It allows uploading and evaluating new tools, and eventually discovering the best combinations of features that can be used for biomarker construction. So again, I wanted to thank my group for helping me be able to present the work of the quantitative image feature pipeline here and the funding agencies. And I then thank you very much for your attention, and I cede the floor to the chair.
Hi. Is there a place for open source packs? And if so, what would it look like? For example, could you imagine a system a bit like the App Store on Android, where it's an open source pack system and different vendors or research groups produce AI systems which can then plug into that open source system?
I think question has to do with open source packs. I know there are small scale pack systems that's open source, but oftentimes, yes, the code is straightforward, but implementation is far more difficult if you try to do that on your own, especially for any mid-size organizations. I do not manage any open source PACS software in my organization at this time, however.
I think, just to comment, you're probably familiar with DCM for CHE, which is an open source data management system for PACS, and you can combine it with a variety of different workstations, which, particularly for research PACS, works out pretty well. I don't know that anybody has combined it directly with pipelines such as the one Sandy just described, but obviously he has a PACS interface, so that is something that could be done.
could I just add this from what we heard this morning and previously in the meeting, there seems to be a clear disincentive for individual doctors, individual hospitals to bring new tech companies on stream because of the difficulty in integrating any proprietary product from an SME into a hospital's healthcare system. If one of the major vendors of PACS systems, say, broke ranks and said, we are going to put our system online, make it pluggable into, would that not provide a real incentive, a kickstart to the system for these SMEs to be able to make plugins or apps that integrated?
So I'll say that actually I think that has already begun. There's at least one vendor that I know of, I won't mention the name, but we can talk about it later, that has a series of plugins and will produce a spec for a programmer to write a plugin for it. Of course, there's all sorts of issues with a physician using a plugin and making some sort of decision based on it, but that's already happened. There's another vendor that has an app store concept for their platform, where they will store a bunch of AI algorithms that you can download to do that, and you can put that on your PACS network and use it as well. So I think that's actually beginning.
I am aware of a couple of companies who provide such service in the cloud. So images are sent from local PACS workstation of the archive to the cloud, and the processed images come back. In fact, one of the speakers at my workshop in Washington, D.C. last week spoke, and the issues were really, even in the United States, different radiology departments are so different. They had to do quite a bit of localization to make it fully integrated into their workflow.
Any other questions? I have a question for Dr. Napel. One of the very reasonable questions that we receive is, what should be the minimal number of patients, your patient sample, in order to start considering developing a radiomic signature? Do you have a number for that? Let's say it's a classification problem to make it easier.
Well, you know, it depends on so many things that actually you brought up in your talk, which are the heterogeneity of the cohort. You know, in the end, when you think about it, you're going to wind up with a very small number of features. You're going to start out with a whole lot, but they're going to be filtered out in various ways that you described very nicely. And then it'll be probably some simple linear model or something not too much more complicated. And so you'd say, well, if I only have 11 features that I have to compute, I shouldn't need more than 100 patients. But is that Radiomix model that I develop in Palo Alto, California, going to be useful for people that are scanned here in Lisbon? I think it's a really tough question to get a good answer to. Probably the closest I can get is to say a lot.
Okay, is there any reason why in your platform you have implemented the lasso method?
Oh, it was done by a student as a student project, and it was the first one that was implemented, and so it's been on the system the longest, but there are a few others that are being written, and there's no restrictions on what it can be. Just very simple, and we have a lot of experience with Lasso because of Rob Tibshirani, who published some of the early work on that, being a Stanford professor and working with him.
Any other questions from the audience? Everyone is waiting for the cocktail, I guess.
I'm only asking because I think the other people are not asking. I just want to take the viewpoint of a lot of radiologists in the room who are just starting out with this Radiomics a venture, and they see the potential of doing so much with it. So somebody here is extremely enthused now, and they're going back and they search, I don't know, 1,000 data sets of a CT of the liver, let's say, and they want to put it through the Radiomix pipeline. And so are we saying at the moment that there are tools out there? Because when we export this space, I'm really lucky to have a team to support us in doing the analysis ourselves, but what I found that it really isn't such an easy process. So to what extent at the moment is there are tools out there that are available, academic or otherwise, that you can actually feed this information in. It will do the radiomics analysis. It would actually do the statistical analysis, which I think is the most complex bit of the process, that without having somebody like a data scientist within your department is really difficult to achieve. So I just wanted a sense of the reality of the people in the room to see what actually they can do now versus what, you know, and if they really want to do that, what sort of infrastructure or people do they think they need to put in place in the next couple of years to enable them to do so?
That's exactly the reason why we will offer tomorrow the hands-on workshop. So,
But if I could just comment on that for a second. I think Dr. Wagner did a great job of sharing her experience of how do you fight your way through this problem. And I think one of the references that Dr. Moon put up for the fMRI community, it became very easy to do fMRI, but without understanding what was in the black box, you got a lot of interesting results that didn't mean very much. And so it really is important to understand what's in the black box and to understand the data and the problem you're trying to solve. And I think so you need to have that kind of expertise in a team that's going to go about this. Until until we get actual products that come out commercially that are trained networks that solve problems in a reasonable way, which is what I think all the commercial people are working.
I have a question. Eventually, the tools that Radiomics would develop will belong to radiology community, pathologists, or oncologists. Who will actually use them?
Who will use them or who will own them?
Who will own them, I should say. Or
Well,
who should own
everything
it?
is open source. And I don't really think about the ownership issues. But who will use them is a good question. I mean, in the endeavors that I'm involved with are more involved in tool development and comparison and just trying to make incremental headway from month to month on the utility of these radiomics approaches. If they actually wind up being used clinically at some point, then most likely some company will take them and package them, and we'll have responsibility for that.
any other questions from the audience?
From a clinical perspective, what we are looking today, what is happening today is that pathology is assuming on oncology a higher and higher proportion of the studies because we need to do molecular signatures, we need to do genetic modeling of the tumors, they become different, metastases become different with different treatments, and there is a clonal expansion of the metastases that do not respond anymore to certain types of chemotherapy that responded before. So that's what we see from the point of view of pathology, and this is an explosion. My answer is Radiomics prepared to accompany this explosion in pathology and give the surrogate markers of pathology, or is exactly the opposite? And for instance, just to give you an example with HEC, with hepatocellular carcinoma, now I think that the wash-in, wash-out concept may be not enough because we need to know what kind of mutations has the the cancer in order to understand better what will be the treatment response or the TTP or whatever. So my question is, how do you see this fight between the biomarkers of radiology and pathology that are exploding on oncology?
Maybe I'll dive in first while Sandy's thinking. So we changed the name to protect the innocent, so that's called Pathomix now. But the problem there is much more difficult. You've already alluded to many of the sources of variants, but just one of the slides I showed, I have 1 million cellular nuclei to look at. A pathologist looks at a very small number of those and makes a determination. My colleague Joel Saltz has shown that if you look at all of them, you frequently make the opposite determination. So the pathomic analysis is really essential to get accuracy, but because there's so many different types of analyses that can be done with a path slide, it's a far more complicated problem. Radiomix is much easier. But there are people working on it, and the two are complementary, right? And I think, to me, one of the most important things is to look at both, because that bridge between what we see in a radiology image of a lesion in an organ, we need to make the bridge to what is seen in the pathology slides to better understand mechanisms. And to me, that's the main reason for applying AI and machine learning is not so much to build tools for radiologists or pathologists, but to use them as research tools that allow us to get new knowledge. And it's that linkage which is important.
Yeah, I would just add that it's not an either-or situation. What we had in the past was a radiologist report, a piece of paper with a bunch of words on it, and a pathologist then looking at the excised tumor, for instance, and also making a report about what they found. And I think that it's not either or. I think that you can make the radiologist's report much more specific by developing good radiomics models where they exist. They won't always exist. And the same thing will happen in pathology as well. So I don't see it so much as a competition as each discipline making itself a bit more quantitative than it had been in the past.
Any other questions before closing the session? Okay. All right, thank you very much, and enjoy the welcome reception.